Perfect. All right, so Rachel works also on the Arctic Network and also on the COG projects, postdoctoral researcher, and going to take us into the world of Pangolin lineages and variants and Scorpio and things like that. So over to you. Thank you very much.
Thank you. So yes, I'm going to be covering Pangolin, Scorpio, and civic today, which are tools that have been worked on within the group. Pangolin and civic in particular have been worked on by Anya and Verity, and Scorpio is something that I've spent more time working on myself. So these are really things that use the phylogenetics to help with genomic surveillance. So pathogen genomics really gives us information about what is spreading in the community. It can tell you about novel imports, and it may give a clue in addition to epidata as to where the imports have come from. It can help to flag unusual or biologically significant changes in the genome, which can help us to identify new variants or lineages of concern. And it can help us to track their spread nationally or internationally. And on the other end of the spectrum, we use this to tease apart transmission chains. So for example, routinely in the hospital and care homes in Edinburgh, we've been using this genomic surveillance and these techniques to try and investigate local outbreaks. And this is something that you can do if you are sequencing in real time. If you sequence as you go, you can analyze, and this is the point where it becomes useful. The results become useful to public health. There were previous strides towards this during the 2014-2016 West African Ebola epidemic, which had nearly 30,000 cases, as it says here. But it wasn't till the very end of the pandemic that real-time sequencing started to be used. And so in that context, it was most useful for helping to stamp out the final chains of transmission. Over the last 2 years, however, there's been a massive global push towards real-time data sharing. So to put this kind of into perspective, in terms of the number of genomes as compared to previously sequenced, since the first genome back in January 2020, by March 2020, there were already more SARS-CoV-2 genomes sequenced than the entire of the HIV database on NCBI. By late 2020, there were more genomes sequenced than a 40-year influenza surveillance program had managed to sequence. And by March 2021, there were more than 1 million SARS-CoV-2 sequences, which is more than any other virus on NCBI. And to date, we have more than 7 million genome sequences that have been shared on GISAID from 195 nations, which is a lot. And nations were sharing these from really quite early on in the pandemic. There's lots of terms that are used when we're talking about variants. We use this word strains, clades, lineages, variants. I'm going to start by clarifying some of these. So first thing is there is only one strain of SARS-CoV-2. Here's a tree made by Trevor Bedford, which shows SARS-CoV-2 in the context of the other beta coronaviruses. And you can see that it's just that one red dot at the bottom there by comparison with all the variation in MERS and SARS-CoV-1. If we zoom into SARS-CoV-1 and SARS-CoV-2, we can see that, again, it looks like there's very little diversity within SARS-CoV-2 as compared to the SARS-CoV and SARS-like coronaviruses. And it sort of clusters down at the bottom with some other bat and pangolin sequences. So zooming in once more, we can see that with its closest relative, the bat sequence, we see that all of the SARS-CoV sequences are separated from that closest ancestor by more than 1,200 mutations. So it's only really when you zoom into just SARS-CoV-2 diversity within the pandemic that we start to see variation. First thing we want to do when we're doing analysis is to be able to bin the diversity into categories. And this is what clades and lineages do. Within SARS-CoV-2, they work on different resolutions though. So clades try to describe broad stroke diversity patterns over years within the phylogeny. And they have a minimum size and persistence criteria. And these are what's used by both NextDrain and GISAID for their classifications into categories. Operating differently from this, a different resolution, are lineages. And these are based off of a set of rules that were first proposed at the, back in 2020. And they try to describe diversity at a level which captures the emerging edge of the pandemic and provide labels and categories whilst they're still relevant to public health. A lineage is essentially a phylogenetic cluster that is associated with an epidemiological event. So this could be an introduction into a new area, it could be an increase in transmission, or it could be something like a new potentially worrying mutation that's occurred within it that we think is likely to be biologically significant in another way. And the system is set up to give labels which represent the hierarchy of the tree. So here, the hierarchy reflects the virus evolutionary history. You've got your first sequences, which were lineage A, and then at a certain point, we started calling them, after a certain number of mutations, we just find a lineage B. And then to stop these names becoming too long, we start having aliases which help us to keep talking about the lineages as they are with their relationship on the tree, but without the long names. Since we started doing this manual curation of the tree to define lineages, there have been more than 1,800 lineages designated. And you can see on the plot that some of these are really quite large. B117 had a huge number of sequences associated with it, but some of them are really quite tiny. So we don't have any of these, we don't have the criteria for minimum size and persistence that you see in clades. And here shown in purple are the designated sequences. So designated means we have manually curated and sort of labeled them with a lineage. And these, we've designated most of the sequences which have less than 5% ambiguity on GISAID. But there's a lot of sequences on GISAID which are new or which have more ambiguities in them. And so if we want to be able to assign a lineage to these lineages that have not been designated a lineage, then we need a tool to do it. So Pangolin was created at the quite early on in the pandemic to do the job of assigning lineages for those non-designated sequences. And this has been made infinitely more accessible to the world because of the Pangolin web application, which has been developed by the Center for Genomic Pathogen Surveillance. And this tool allows you to drag and drop your sequences, and then you can export or sort of link out to Microreact and to COV lineages and so on. They passed the milestone of 1 million sequences, which have been investigated on their website in March 2021, and we're now nearly a year on from that. So it's had a lot of use. So here we have a picture of the global tree with our manually curated lineage designations. And in the first iteration of Pangolin, what we tried to do was create representatives of each of those lineages. And we would pick those representatives, and then we would try and use maximum likelihood to find the best location for an input query sequence on the tree, and maximum parsimony to infer the most likely lineage based off of that. Now, this didn't scale very well. So with the massively expanding number of genome sequences that were being added onto the database, we found that by June 2020, it was just not going to scale anymore. So Pangolin 2.0 introduced Pangolin, which is a it's a machine learning algorithm written by Emily Scher, which instead trained a model, which in this case is a decision tree, to try and use the information from the manually curated lineages and use that information to infer the lineages for new sequences. This was much, much faster. We can now process millions of sequences in a few hours. It takes all diversity into account. Because it's a decision tree in machine learning, homoplazy should be uninformative. And it has proved relatively robust to ambiguities. You can see that when sequences stop being correctly assigned because of ambiguities in the sequences, first the parent is assigned. You have small numbers that are incorrectly assigned, but then they do tend to eventually just fail to map. But most of the time, if you either get your lineage or you get the parent of the lineage that would be correct. But the problem with this method is that it is overfit. And this means that the accuracy does drop off as the virus evolves beyond the known diversity. And this is a known problem. And so within Pangolin 3.0, we introduced alternative inference engines. And the biggest change really was the introduction of Usher Mode. Now, Usher works much more like the first iteration of Pangolin. It places sequences using fast parsimony onto an existing tree of designated sequences, but it uses placement with the tool Usher and therefore scales more robust and therefore scales better than the original method did. Usher tends to scale more robustly with increasing genetic distance, but it does suffer being considerably slower than Pangolin. So it's been a very helpful alternative inference for when you're wanting to query a smaller number of sequences quite carefully, but is less suited to recalling the entire database within a reasonable time frame. In addition, as variants of concern and variants of interest have cropped up in over the last year, we added Scorpio as an extra tool within Pangolin to validate lineage calls for variants, and I'll come to variants now. So what is a variant? A variant can just be a mutation, like a SNP or an insertion or a deletion, but typically what we mean when we talk about variants are viruses defined by these mutations. So a virus with a particular set of, or combination or constellation of mutations, which we think have biological significance. And we talk about variants most when we're talking about variants of concern, variants of interest, or variants under investigation, which are listed by WHO. And for the variants of concern, we've now got 5 variants of concern with names. And these are the constellations of mutations which represent those variants. There's quite a lot of mutations there on those lists. So what Scorpio does is it carefully checks each of those mutation sites within the genome and it types them. And then it just uses it scores how many of those sites have got the reference allele, how many have got the alternative allele, how many were ambiguous. And on that basis, it makes a decision about whether or not it should be classified as that variant. It's worth noting that lineages are not equal to variants. And as an example of this, the alpha variant of concern, which was originally represented by lineage B.1.1.7, there is another variant which was investigated, which was B117 plus this extra mutation E484K. And this mutation has occurred multiple times on the tree. So within just the B117 lineage, it's occurred independently 25 times, more than 25 times. And this is what's known as homoplasy, because mutation occurs independently more than once across the phylogeny. Another early variant was this mutation DUG in the spike protein, which occurred prior to the large outbreak in northern Italy. And this has been known to occur, well, it now dominates most of the sequences across the world, but it has been, if you look at the tree, it's shown that it's occurred multiple times across the tree. as has another mutation at the same site, which converts the D amino acid to an N instead of a G. Happily, in the case of this mutation, whilst it increased transmissibility, there was no evidence of any link to disease severity. And similarly, we had a case of this mutation, which worked in a similar way, Q613H in the A23.1 lineage, which first occurred in Uganda. And this mutation, again, seems to have had a similar effect of increasing transmissibility. So moving on to slightly different sort of question, we're looking at investigating outbreaks. Collaborators over at the Royal Infirmary in Edinburgh from quite early on in the pandemic wanted to be able to get phylogenetic analyses and reports based off of their sequences that they were generating within the hospital, and to use them to inform about local transmission events. And at this stage, early on in the pandemic, Anya was manually getting the relevant sequences. She was running Pangolin and writing reports on the specific outbreaks, using things like the lineages, but also trying to convey quite nuanced information about how things that look like a large polytomy of identical sequences on the tree were not necessarily equal to transmission and could not be used to say for definite there was transmission going on, because you could see other sequences globally clustering with them. This process wasn't real time because Anya was very busy. And as another example, she also was asked to help a collaborator, Placide, over in the DRC to investigate surveillance of SARS-CoV-2 in the DRC. And to do this, it's not a trivial analysis. She had to download from GISAID, run QC filters, downsample global sequences. You had your masking of the UTRs in the alignment, Pangolin, running alignment, tree building with the sensible outgroup, annotating the tree, Parsimony reconstruction of ancestral states, and then looking at introductions manually and with Parsimony for an upper bound. So none of it's very hard, but it's quite time consuming, and that added to how long it was taking to get these results. So it wasn't real time, but the same analysis could be done locally and quickly with the right tools. And therefore, Anya and Verity wrote civic, which is a tool to do just that. It helps to investigate local outbreak and rule out transmission chains and inform contact tracing efforts. So it essentially works by, like in theory, taking a catchment on the global phylogenetic tree of sequences that provide the context for your data set that you're investigating. And initially this was done using methods on an actual global phylogenetic tree, but as the tree has grown to 7 million sequences, we instead now use a pseudo tree aware SNP distance method written by Ben Jackson called Go Faster to pull out these catchments of sequences. Once we've got the catchment, civic builds a local tree, and then it provides a report which allows you to investigate and sort of explore the data, so you can see all of the available metadata for the sequences that you have input and the ones that are related. You can look at local trees, which they sort of can collapse nodes and expand nodes on those trees to look at information that may or may not be relevant. And you can color the nodes based on any metadata field that you want. You can start looking at clustering geographically, whether things cluster in space or whether they cluster in time. So if you've got repeat infections or if you've got new outbreaks happening. And you can also use civic to look at their spatial context for things. Snippet allows you to zoom in and look at the mutations, specific mutations occurring within your sequences to help tease apart the relationships between them at the SNP level. And that same analysis, which took so long for the DRC, can now be run with a simple one-line command and will provide all of the same results. So civic is now being used routinely by much of COG UK and other places across the world, which has been great. So I think I'm actually going to end there with that. I've touched on Pangolin, Scorpio, and civic and how they've been used to provide analyses based on phylogenetic analysis. And I'll just end by saying thank you to all of these people. There's massive team effort that goes into these sorts of tools and keeping them maintained. So thank you.
Fantastic. Thank you very much indeed. That was really, really helpful guide to some of the amazing tools that you and folk have developed. Really appreciated by me and many people across the world, I think. So hopefully get some more customers from this course. So there are some questions in the chat, so let's go straight to those. So Lays has asked, why do we use a B lineage as a reference, not an A lineage?
For the tree?
I guess so.
So I think there is a simple answer that the first genome sequenced was, I think, I'm going to get this the wrong way around, but the first genome sequenced we don't actually think was ancestrally the oldest sequence. So there's a difference between the reference used for alignment and the reference that is the global reference. So the one that we think is most ancestral to all the sequences is the one that's used for tree building, whereas the one that was the first genome sequenced is generally used as the reference for alignment and as the general SARS-CoV-2 reference.
And does that matter in any way, kind of the choice of reference?
There are two sites that are different, two mutation sites that are different, two mutations between them. So in most cases, it will make very little difference.
Okay. And I guess you can play with the
There are a few places where the nuance of which one you've chosen will matter, but most of the time it's for trees. I would recommend using the ancestral reference rather than the that would
be the best if that was.
That would be the best, yes.
Okay, all right, makes sense. Okay, so Kadim asks, is it possible to deposit your data in Pangolin and view the tree directly in Microreact?
I think pretty much. So what happens is you drag and drop your data onto the web app, and then the web app will give you a link out to Microreact to view your sequences afterwards, I believe. It's not going to give you the same results as civic. It would be a different analysis.
Yeah. Yeah, I think you can, can't you? You can click on and see that lineage in Microreact. Next strain or next clade does something like that as well, doesn't it? You can.
Next clade definitely does something like that too.
Yeah. Okay. So a question from Li Ching is, how accurate for the phylogenetic topology structure using the Usher method? So how accurate is Usher with maximum parsimony compared to IQ tree, which I guess is.
So in the context of SARS-CoV-2, the Usher method and the maximum parsimony method has proved really quite reliable because there are so many sequences and there is not that much diversity. And the way that Usher is incremented, they're able to optimize the tree as well periodically, which leads to a very high quality tree that's underlying the Usher method. IQ tree would not scale to a tree with millions of sequences in it. You could not use IQ tree to do this.
So practically it's
not an alternative.
Do I recall that wasn't there an issue in the past? I don't know if it's still a problem, but does Usher correctly deal with those n-masked regions associated with tile dropouts? Did I recall that it used to struggle with the correct placement based on ambiguities? I may have got that wrong.
So I think Usher ignores ambiguities. The tree ignores insertions and deletions. It's a mutation-only based tree. And there are, I think, occasional cases where that leads to less than ideal placement. But the fact that they're able to optimize the tree periodically means that they can improve it gradually over time, I guess a little as well, because if you've got high quality sequences that you're also placing on the tree, then those would hopefully pull your sequences with slightly more ambiguities back into the right place. I could be wrong about that, but that's my understanding of it.
certainly that Usher method does seem to be incredibly useful in terms of making some of these analyses tractable. Because I mean, it's probably just worth reflecting on. 7 million sequences is just extraordinary by any measure. I said it in my opening slides. I think that's more genome sequences for SARS-CoV-2 than there are genome sequences for literally everything else in the entire database. Is that fact correct?
So definitely there's more, like 1 million sequences was how many virus sequences there were on NCBI. Right. So there was 1 million of those. So we've got 7 million of these.
There could only,
I think for comparison
there was only 1 million or so bacterial whole genome sequences as well, weren't there?
I can repeat that, but I don't know.
Probably a handful of kind of big eukaryotes. So that, it's just amazing. It's amazing to think that in parallel with the evolution of the virus, in parallel with the collection of these genome sequences, had to evolve the methods in sync. Really, it's scale
of the data has been one of the biggest challenges of doing these sorts of analyses.
Yeah, I think the tools that you described are really helpful, because it's clearly, I think for most folk out there, it's not tractable to try and go to a de novo tree, of everything from scratch each time. And so these tools you mentioned, Pangolin and Civic, Usher, are just incredibly helpful for making it possible to look at your context, whether that's, a hospital, a country, something like that, in the global context, which is obviously where a lot of the interesting information comes from.
Yes, thank you.
Well, thank you. I don't see any additional questions, but maybe if folks do have more questions, post them in the chat, and we might be able to get to them before the end of the session. Well, thank you very much, Rachel. That was fantastic. We'll